Thank you. Go ahead.
Thank you so much, Becky, for the introduction. I am Rosario Miranda. I am a PhD student at ENS in Paris. And today I will tell you a little bit about the dynamics of communities and equilibrium on an evolutionary theory of island biogeography. The theory of island biogeography, as developed by MacArthur and Wilson, tried to explain richness equilibrium in islands by two simple processes, immigration and extinctions. And since the 60s, this theory has been really useful in ecology, principally for those that search to explain community dynamics in ecological timescales. The theory of island biogeography also explains how area and distance from the source of species can affect this state of equilibrium. For example, islands that are large and near the source of species achieve an equilibrium state with a higher species richness, and islands that are close, that are far from the source of species and smaller. Since the 60s, we know, nevertheless, that speciation is one of the main mechanisms that drives and builds communities on islands, that forms endemic species on islands. In particular, there are two mechanisms acting on islands, anagenesis and cladogenesis. And taking into account the commonness and frequency of these two mechanisms on islands, it's really important that we extend the theory of island biogeography to these mechanisms, two mechanisms of speciation, if our aim is to study community dynamics in an evolutionary timescale. That's what we did. No. There has been before some approximations, some studies that try to incorporate speciation on the theory of island biogeography in various ways, but nevertheless, there has not been a model that can predict the dynamics of an entire insular community by taking into account immigrant species and endemic species. And there has not been a model also that can predict a state of equilibrium or predict when this equilibrium will be achieved and if this equilibrium will be achieved. This is what we did now. I will introduce you now very briefly to the families or models that we developed for this project. The A model adds to the basic theory of island biogeography anagenesis. So includes three processes building island richness: immigration, anagenesis, and extinction. The AD model builds up on the A model to incorporate the effects of area and isolation on these three processes. So we know for the theory of island biogeography that immigration decreases with isolation and extinction decreases with area. On top of that, we add the assumption that anagenesis should increase with isolation. And these two models can predict the number of species of immigrant and anagenetic species on an island through time. The C model, instead of anagenesis, includes cladogenesis to the theory of island biogeography, and the CAD model adds to the C model the effects of area and distance into the three processes that conform it. And in particular, the assumption that we add here is that cladogenesis should increase with area. And these two models predict the richness of immigrant species and cladogenetic species through time. And finally, we have the AC model that it's a combination of the last two models that incorporates anagenesis and cladogenesis as processes driving island richness. So we have four models: immigration, anagenesis, cladogenesis, and extinction. And finally, the AC-AD model builds up on the AC model to incorporate the effects of area and isolation on the four processes, as I explained before. And this model has the ability to predict the number of immigrants, anagenetic, and cladogenetic species through time. We also were able to predict the conditions for equilibriums for each model. Here in each row, I am showing you first the simple models, the models without the effects of area and distance. And in each column, I am showing you the type of species the model contains. For the A species, we can conclude that the equilibrium will always be achieved. So given enough time, an island under this model will always achieve an equilibrium state. For the C and AC model, the condition of equilibrium is determined by the dynamics of the cladogenetic species. And in particular, an equilibrium will be achieved only if the rate of per lineage cladogenesis on the island is strictly less than the rate of extinction rate on the island. In any other case, an equilibrium will be achieved. And we know also that the equilibrium that is achieved will be stable, meaning that the community can come back to an equilibrium after a disturbance. Here I am showing you the exact same information, but for the models extended for area and distance. In the first column, in the first row, sorry, we can see that the AAD model again always assumes or predicts a state of equilibrium. And the CAD model and the ACAD model, the condition again for equilibrium is determined by the dynamics of the cloud-genetic species, and the condition is pretty much the same as for the simple models, but here the area of the island is modulating the condition for equilibrium, meaning that the area has an effect on the possibilities of achievable of equilibrium of an island. And in all of the cases here, the equilibrium is again stable. I am showing you here a little demo of what our models does, in particular the ACAD model, the model that includes anagenetic species and cloud-genetic species. On the top row, I'm showing you, I plug in the model some parameters that fulfill the equilibrium condition. And I'm showing you the dynamics through time of immigrant species, anagenetic species, and cloud-genetic species. We see that when the condition of equilibrium is met, all of the parts of the community have an asymptotic behavior. They saturate, they achieve an equilibrium state. In the bottom row, the parameters are pretty similar, but I modify them a little bit so the equilibrium condition is not met. And here we see that the dynamics of immigrant achieve an equilibrium, the anagenetic species are about to achieve an equilibrium, but for the cloud-genetic species, the growth of species richness is exponential, explodes. There will not be an equilibrium in that island. So we proceed to test our predictions empirically. For this, we use the Valente dataset of insular bird species. That it's a dataset that includes species information about species richness on archipelagos distributed worldwide of birds. And it also includes information about the origin of the species, meaning if they are in the island by colonization events or if they emerge in the island by in situ speciation. So to fit our model to this data, we follow these steps. First, we classify the species into colonizers, yes, but also into anagenetic and cloud-genetic species in particular. We optimize our model parameters using this data, using all of the islands, which means that we carried out an optimization algorithm that tried to diminish at most the discrepancy between the predictions of the model that we are using and the observed data. And three, with the optimized parameters that we obtained, we predicted the present date richness information on each island. We did this for all of the models and we compute the AIC. Here, each row contains first a simple model and after the model that is extended to include the effects of area and distance and the processes. And the delta AIC is computed by row. I want to show you two things. First, that the best model in our case for our data is the AAD model. That is the model that includes anagenesis as forming endemic species and the effects of area and distance, followed by the CAD model. And I also want to point that the delta AIC is first significant and after is really high for all of the models, which means that adding area and distance improves the fitting of our model by a lot. Here I am showing you the comparison between our predictions and the empirical data for the best fit models. First, I am showing you the information for the richness of immigrants. Here you will see a link pair of points. Each link pair of points correspond to the same island, and the observed richness on immigrants is shown in orange, as found by Valente et al. Present day richness is shown in orange, and our predicted present day richness is shown in green for the AAD model again. Here I'm showing you the same information, but for endemic species, the AAD model assumes that the endemic species are anagenetics. So it's the same thing. The linked pair of points represent the same island. The orange dot represents the observed richness, and the predicted point by the AAD model, it's the green dot. And the link of the gray link, it's like a represents how accurate are our predictions. If the gray link is high, it's a bad prediction. If the link is small, it's a good prediction. Finally, here I am showing you exactly the same, but for the total richness of the island that I compute by adding the richness of the sub communities, colonizers and endemics. And for the most part, we can see that it's a really good fit, the one that we obtained, that our model can pre-accurately predict the present day richness on islands. So now we can check with our optimized parameters if our islands met the equilibrium condition, meaning if our islands will achieve an equilibrium given enough time. So for this, we pick the two best models. For the AAD model, the condition of equilibrium is trivial. An equilibrium will always be achieved. For the CAD model, the model that includes cladogenesis on the theory of island biogeography and the effects of area and distance, we check the condition there with the optimized parameters, and all of our islands fulfill the condition of equilibrium, meaning that they will arrive to an equilibrium given enough time. We also use the parameters derived from the machinery, the inference machinery of Daisy. And for these parameter combinations, all of the islands except for Galapagos and Hawaii satisfy the equilibrium condition. So we know that our islands can arrive to an equilibrium. So which islands are on equilibrium right now given their age? So it's a question that we can also answer very easily with our models. And our models predicted that 19 out of the 39 islands that we utilize are already on equilibrium right now. So I want to conclude telling you that we develop an evolutionary extension of the theory of island biogeography that can predict the dynamics of entire insular communities, including endemics and colonizers. That for our data, the best fit model was the model that includes anagenesis and area and distance. And of the analyzed islands, all of them except for Galapagos and Hawaii meet the equilibrium conditions, and 19 of our islands are predicted to be in equilibrium right now. I want to thank my incredible director, Hélène Morlon, and my beautiful lab for all of your support, and thank you, Becky, and the Organization of Evolution for this opportunity. Thank you so much. I will return to the conclusions, but I finished.
Thank you, Maria, for a wonderful talk. And we do have a few minutes for questions. So feel free to raise your hand and then we can give you the option to unmute, or feel free to drop them in the chat and I'd be happy to relay them. If I could figure out my Q&A options. Perhaps while people are conjuring up those ideas. Oh, here we go.
Okay.
Maria,
Can you read it for me, please?
Yeah, I was just going to say, I'll read it so that
Awesome.
everyone else can also hear. So Darren in the chat says, Maria, very interesting model and talk.
Thank
Perhaps
you.
I missed it, but I'm wondering how archipelagos might affect the model. Nearby islands might accelerate speciation and or cladogenesis rates.
Yeah. That's one of the basic points of the island biogeography. And the point here is that we are, our object of study is birds, and birds are known to be very mobile. And it's known that the cladogenesis in principle, it's the cladogenesis rate is higher in archipelagos than in simple islands. But yeah, we have this particularity that we are talking about birds, and maybe the result will be different, or there will be a higher effect of our study if we talk about more terrestrial beings. But yeah, I mean, in the Valente paper of Nature, they
Diego, why don't you
yeah.
go ahead and get your
Sorry.
slide set up? Sorry.
Yes. They just mixed islands and archipelagos because they are talking about Perth, so we did the same thing. But yeah, uh,
All right. Well, thank you for that, Maria. I appreciate that. We're at time now, so I encourage you to keep engaging with the Q&A. It's a great way to continue the conversation. Um, but with that, we have our next speaker, Diego. Take it away, please.
Sorry about that. Hello, everyone. Thanks, Becky, and thank you, everyone, for being here. It's very exciting to share my research in the Ernst Mayr Symposium. Thanks for the organizers for accepting my talk. I'm Diego. Can you see my screen? Hopefully you can. Yeah. Okay. My name is Diego. I'm a postdoctoral fellow working with Jeremy Bulliu at the University of Arkansas. But what I'm going to show today, what I'm going to share today, I've been working with this piece of research until a semester ago at Louisiana State University with Laura Lagomarzino. And today I'm going to share about mountain biodiversity. This is specifically for plants, this plant biodiversity. You can see the map. The map is a species richness of plants across the globe in coincidentally, or maybe not so coincidentally, species richness for plant peaks where mountains occur. And we have mountains in Southeast Asia between Europe and Asia and South America. And within South America, I'd like to highlight the tropical Andes, not only because of its territory, its vast territory along the western side of South America, but also because of the richness that these mountains are home to. Around 10% of the global flora occur in the Andes, and you can see that the species richness is not homogeneous. It's quite heterogeneous, with the northern part of the Andes, or the tropical Andes specifically, having even more species. And with that, a few representatives are the Puya Raimondii on the left, or the Queen of the Andes, or the frailejones from the Spelitine tribe on the Paramos. Puna on the left, the Puna ecosystem, Param ecosystem on the right. And most of the species, like around half of the species occurring in these mountains, are restricted to just the Andes. And this species-rich flora has originated pretty recently, within around 1.5 million years ago. If we compress the Earth's history is within a single year, that's pretty much 5 minutes ago. So that's a very recent diversification. And in this case, for the Andean bluebottles, in the case for many lineages in the Andes, just a single lineage arrived to the Andes and quickly diversified. So for my dissertation, one of my main questions was which evolutionary processes are driving this rapid recent diversification that we're seeing in the tropical Andes. So with that, I'd like to focus first on a single clade point of view. I'm going to talk about the genus Brachiotum, which occurs along the tropical side of the Andes, you can see, with species peaking between southern Ecuador and northern Peru. This genus belongs to the Melastomataceae family, which is part of the meadow beauty family, if you're familiar with the meadow beauties here in North America. Brachiotum comprises 55 species, and all these species, and that's pretty unique for a Brachiotum within the family, all these species have this morphology where the petals form a tube, so hummingbirds can pollinate the flowers. And not only that, they produce nectar. All of those traits are pretty unique within the family. But despite that, the phylogenomics or the evolutionary history of Brachiotum is pretty complex. If we use plastid loci, we have that Brachiotum is not monophyletic. If we use nuclear data, that same nuclear data set applying different methods, we're going to find out that Brachiotum can be monophyletic or paraphyletic, can be recovered as monophyletic or not. So in the presence of this diversity of topologies of evolutionary histories, we have that single and individual loci have different histories compared to the lineage. And that is the case of gene tree discordance when we have the evolutionary histories of gene trees and species trees differ. There are multiple sources that drive gene tree discordance. One of them is hybridization that has been hypothesized for Brachiotum. To test that, I applied a phylogenetic network. I inferred a phylogenetic network with nuclear loci. And in this case, we found that there were at least 4 introgression events, not only within Brachiotum, but also with lineages related to Brachiotum, closely related. Just a single lineage was placed within Brachiotum, and this single lineage was found to be a potential hybrid when we assessed the allele divergence and locus heterozygosity across all the nuclear loci. And all these couple pieces of evidence are also in alignment with morphology. Morphological evidence says that it can be a recent hybridization event. This is in the case of a single locality. The three species or the three taxa that you're seeing occur in the same single locality in southern Peru. So we have one species of Brachyodon, we have one species of Chetogastra, and we have a certain intermediate taxon that has some traits that resemble Chetogastra and some traits that resemble Brachyodon. So again, the hypothesis is that this is a recent hybridization event, and maybe that is what is affecting the whole monophily and paraphyly of Brachyodon. And again, this recent hybridization event occurs within a recent diversification in general scenario for all the species of Brachyodon. Brachyodon, similar to the Andean bluebonnets case that I presented earlier, diversified within 1.5 million years ago. Again, that's 5 minutes ago. If we compress the whole Earth's history within a year, that's super recent. And Brachyodon, in this case, originated at the southern portion of its distribution by the Central Andes. And in that case, in that context, the geological and climatic context is that within 1.5 million years ago, pretty much we were seeing already the elevation that we're currently seeing. So mountain orogeny was pretty much already built, and it was on the last leg of mountain building. However, climate was still, it was basically changing a lot, specifically talking about the Pleistocene glacial cycles that pushed the elevational distribution of species up and down, higher and lower elevation, with caused a repeated cycle of isolation and secondary contact. This leads to thinking me. I started thinking about this, and pretty much elevation describe well what has been happening with the distribution of species, but also higher ecosystems tend to comprise only lineages that are very recent, that have diversified very recently. So that, what the pattern that I saw with gene tree discordance in Brachyodon led me to think what happens across the Andean flora. So in this case, I thought maybe younger ecosystems at higher elevations that have had more recent diversifications have undergone higher gene tree discordance. So for this, I compiled 11 datasets, phylogenomic datasets across the angiosperm tree of life to complement the data that I generated for Brachyodon. And in this case, my approach to compare empirical datasets to assess gene tree discordance, I apply the quartet sampling scores approach, which measures gene tree discordance and compares the inferred topology and its underlying alignment, the alignment that produced that topology. So in this case, we have two scores. I'm going to present two scores. QC, quartet concordance, that finds the support in the frequency based on the frequency of the reconstructed topology, while QD, under the presence of gene tree discordance, is going to measure how imbalanced are the frequency of the alternative topologies. I showed you for Brachyodon that there is a diversity of topologies. QD is going to measure the alternative topologies and the frequency. So if there's an imbalance in the frequency of alternative topologies, QD is going to be lower. So with that, my hypothesis was that both QC and QD are going to be decreasing with elevation. What we found is that QC had lower values across all lineages, and in some cases, negative values, meaning that the reconstructed topology was actually not the preferred, or quote unquote, the real one, and one of the alternatives was preferred. And these lower values of QC occurred at lower elevations, which is totally opposite to what we were expecting. Brachyodon is in the middle, so there are worse cases than Brachyodon, unfortunately. For QD, we have that there are lower values across all lineages as well, and that means that again the alternative topologies are imbalanced, the frequencies are imbalanced, with one of the alternative topologies being preferred. And in that case, lower values of QD were increasing with elevation. So, okay, that's aligned with our expectations. Brachyodon, again, in the middle. But what is originating these patterns of QC and QD, and we were finding completely different for QC, completely different to our expectations? Here, I want to say that there are multiple evolutionary processes that drive gene tree discordance and are driving these two facets of gene tree discordance that we're measuring. There are gene copies, whole genome duplication that both are frequent in plants. There is recombination, there is rate heterogeneity, etc. There are multiple sources that are causing, the interplay of all those sources are causing multiple scenarios. However, I'd like to focus here first on two main sources: incomplete lineage sorting and gene flow. And why? One, because that's what we understand the better from phylogenomic data sets because, two, that's what most pipelines incorporate in the methods that we're applying to study empirical data sets. So with that, one of my hypotheses is, again, across the myriad of scenarios that the interplay between these sources are generating, one scenario may be consistent with incomplete linear sorting, having less of an effect on the smaller populations that are occurring in isolation at higher elevations, thus QC increasing with elevation. Again, that's one potential scenario. Another one can be that QD being lower with elevation is consistent with hybridization. Any introgression from between species, from one species to another, is generating this imbalance in the alternative topologies, thus generating QD values to be lower with elevation. Again, this is a complex scenario. I'm trying to make a couple of hypotheses of scenarios that are they're having potential for the diversification of Andean clades. However, again, there is an interplay of multiple sources. And with that, I'd like to say pretty much that plant evolution in the Andes has generated evolutionary scenarios more complex than the current methods that we are applying to study empirical data sets, empirical cases of plant diversification in the Andes. Maybe the approaches are not complex enough to explain all of that and to incorporate all the complexity in the evolutionary history. With that, I think I have good time. I'm good with time. I'd like to thank you all again. Thank you to the Lagomars in the lab at LSU, and thank you everyone that supported me and all the institutions that support my research as well. And thank you to SSB for having me today.
Thank you, Diego. And again, please feel free to raise your hand if you have a question and or submit in the chat, and we'd be happy to relay. In that case, I have a quick question, Diego, is given the complexity of, well, the fact that I think you put out a good point is the more we look into it and the more data we have to look at these complex systems, we're realizing how much more complex they really, really are, and what goes into it. And so how do you feel like, or where are we with kind of this interface of, yeah, we have a lot more data, and now we're realizing we don't really have the tools or models to really capture that. And so do you have any recommendations on how to tackle?
That's a great question, and I've been thinking about that. And I think empirically,
Yeah, right?
yeah. Well, just briefly sharing, the first chapter of my dissertation was looking at the evolutionary history of Percutum, and you saw that I have a diversity of phylogenies, and none of them was well supported. And at some point, I was like, I don't know if I feel confident using any of them for further analysis. And I've been thinking about that, about what can we do with the data that we have, despite having more data. There is not much that we can do so far. I've seen that there are recent advances in incorporating recombination, for example, and incorporating gene copies. There are methods that are incorporating both. I think the challenges, despite having some approaches and some advances in methods, is also, the couple challenge is that in my case with plants, we work with herbarium specimens that sometimes the DNA quality is not the best. So it's the
Sure.
combination of having the empirical challenge of using what we have for the samples and the challenge of having the DNA quality and the methods maybe not incorporating everything. So
Multiple levels.
yeah, multiple levels of challenge. Briefly, let's do the best that we can.
Yeah,
I
best we
think,
can with what
I
we have.
think yeah, on top of that, and I think that my answer is pretty much describing what we currently have, is that I presented a phylogenetic network. The computational time to also infer a phylogenetic network is very challenging so far. And we need to reduce our data sets to be able to infer a phylogenetic network. So I'm just saying there are multiple levels, again, of challenges. And I think maybe as a community, we need to find out together what are the best approaches. I'm trying to highlight here that this is not only a single lineage pattern, it's across the
Yeah, right. Yeah, you're talking to a larger phenomenon that I think a lot of people are coming across as
Yep.
we're collecting these types of data to still ask these
Yep.
fundamental questions. So
Yep. Thank you.
fantastic. Well, I didn't mean to take up the most of the Q&A session. So if someone else has a quick one,
Mhm.
but
Yep.
otherwise, I think we're at the top of the next talk.
Okay.
So
Thank you, everyone.
thank Diego again for a wonderful presentation. And then we have our next speaker. You want to get your slides set up? Oops, sorry.
Yeah, sure. Cool. I hope my slides are visible.
Yep. Looking good.
So, hello everyone. I'm Karunakar. I'm a first year graduate student at the Center for Cellular and Molecular Biology in India. And here in the symposium, I'll be presenting a case study on the miniaturization of a genus of centipede called Asanada. So, just to give a quick start, there are two contrasting phenomena that we know about body size, and one of which is a maximizing trend, which is actually Cope's rule, and which tells us that there is a macroevolutionary trend towards increase in body size over evolutionary time. And the second one is a minimizing trend, which is miniaturization, where lineages basically shift to smaller body size over evolutionary time. And keeping this in mind, I wanted to study something related to this. And I like centipedes and I like studying centipedes. And I didn't have to go any far to observe this phenomenon of miniaturization. It was right there, the next door. So mostly study group of centipedes, or a family of centipedes called Scolopendridae, and within this family there are few groups such as Asanada, which are roughly around 25 to 30 millimeters in size, whereas they are nested within a large-bodied centipede called Scolopendra. And these particular centipedes are actually 4 to 5 times bigger in size as compared to Asanada. So based on this, I wanted to look at what are the drivers, what are the patterns in body size evolution, and I wanted to first check what all information is actually there in this regarding this particular group of organisms. And actually, not everything, but what I would like to say that there is a very limited amount of information that was already present. So these are profiles of a micropredator, like it belongs to the family Scolopendridae, it belongs to the tribe Asanadini, and there are key species, 3 to 4 key species that are present in this particular genus, which are described from the Indian subcontinent. And I wanted to look at this particular lineage from the perspective of, or from the facets of DNA or morphology or geography or ecology or their natural history. So we just knew that they are mostly found literally anywhere and everywhere. And that's why we had a tough time looking for them. They are very discreet, and we actually had to go to multiple field visits to actually find this lineage. And coming to the field sampling efforts, we went on a voyage around different discontinuous or continuous landscapes in the Western Ghats and Eastern Ghats of India, as well as Andamans. And we created a matrix of 61 individuals for as our primary data, and 2 individuals which we retrieved from the published data. And the morphological characters that we could score were 17 in number, and the molecular data that we generated was 2 mitochondrial markers and 1 nuclear markers, and and we created a combined data set of 1650 base pairs, and we did maximum likelihood as well as Bayesian inference approach for tree building. And we wanted to test this particular hypothesis based on Joshi and Karanth 2014 paper, where Asanada is nested within this larger bodied taxa, Scolopendra. And since Scolopendra is also paraphyletic. So we try to look at the morphotaxonomy, and as described in the paper Scolopendridae of the Tekken by Janghi and Das, there are total 17 characters, morphological characters that were scored based on this reference. And all these morphological characters basically tell us that there are 3 species or 3 clusters. So we try to validate this based on our data set. So whatever data that we collected from peninsular India, we try to validate it. And to my knowledge, it was just three different characters which actually showed some kind of variation, and because of which I could delimit the morpho species based on and this from an ordination analysis, I could find out that there are three clusters, three non-overlapping clusters, although the spread is pretty high, but there are three major clusters that our dataset yielded. And from literature, I also know that there's evidence of pedomorphosis, which is retention of juvenile traits in adult body forms. But this is also notorious to actually uh, mess with the characters or mess with the diagnostic characters of a particular lineage. And it makes it difficult for us as well to correctly declutter or decluster it morphologically. So we tried coming to the perspective of DNA, and we built a phylogeny and passed it on to different species discovery models. And then we tried to estimate what do these species discovery tools tell us, what is the number of species that they can mine. And from this particular data set that we had, we could demarcate 10 distinct lineages, although the number of species that are yielded by our species delimitation tools is quite exaggerated, and we wanted to go through a very conservative approach. So we tried to look at different distinct units of discontinuous mountain chains and how they are occupied by different lineages. And we tried to collate all of those into one, and we tried to come up with a species hypothesis. So based on whatever we studied till now, so that has significant evidence that Asanada as a lineage is morphologically cryptic, because there are many more independently evolving lineages within this entire genus. So I also wanted to know where all is it actually present. I wanted to get all the data out of the internet. I wanted to work with that data, but to my surprise, it's not present everywhere, although it shows that it's present from the plains of Africa to the rainforest in Queensland, but the amount of data that is actually there is just two molecular sequences apart from whatever primary data that we have in our study. So we try to build whatever we can, and the next step was to estimate how old this lineage is. So we tried doing it at a family level, and we had our phylogenetic backbone for 60 species within the same family, and we used optimized relaxed molecular clock and GTR plus gamma substitution model, and also used a log normal prior using existing fossil data. And we constrained the monophily based on respective clades. So based on Joshi and Karant 2011 paper, we tried to rebuild this entire phylogeny with whatever data that we had collected or generated over years. And this is the family level representative of the entire Scolopendridae family. And there is a zoomed in representation of what the time of splits across this across our clade of interest actually is. So we can clearly see that our clade of interest, or the lineage that we are working on, is pretty old. It is roughly 152.7 million years old. And literature also suggests that their body form, or the body form of chilopods, hasn't changed much over time. So we can definitely call this a living fossil, as this is a lineage which is pretty old. And I wanted to use all this data together to stick to my core question that what is the dynamics of body size as a trait across this entire family, because there's a really tiny lineage within a large bodied clade. So I wanted to look at that aspect as well. So this is a clear methodology which I followed. I had my input as a time calibrated species tree, and my trait data in terms of body size. So I tried to fit different models like Brownian motion, OU, or early burst. And all these models come with their underlying hypothesis, such as whether my trait is walking across evolutionary time in a random fashion with increasing variance, or is it following an optima or an optimal value which is pulling my trait. And all of this combined, I tried to fit which model I tried to estimate which model fits my data the best and explains it the best. And using these models, or using the model parameters from the best fit models, I tried to estimate what are the ancestral states of the ancestral nodes in this entire phylogeny, or our concerned divergence dating tree. So for that, we needed a phylogeny, which we already have, and the morphometrics, which is the measurement of body size as a trait. And we wanted to reconstruct its evolutionary past, as I had mentioned, and to stick to the same core question: what is the dynamics of body size evolution across this entire family? And we found that the ancestral state reconstructions supports Brownian motion mode, which tells us the traits are evolving neutrally, or the traits are basically randomly evolving through time. And this is a ancestral state reconstruction which tells us that smaller body form evolved once in the entire family, and this is the smallest of all, with independent shifts to higher body size or larger body size multiple times, but most of it remains very consistent throughout the entire phylogeny. So these are the dynamics of body size evolution as a trait, and we can see that our clade of interest, the clade of interest that we are working on, has the least body size, and the close relative, which is Scolopendra, has a really large body size. And most of it remain considerately similar throughout this entire phylogeny. And I also tried estimating the phylogenetic signal for this. The Pagel's lambda is 0.737, and the Bloomberg's K is 0.441, which suggests low to moderate phylogenetic signal for this trait. And I wanted to get all of this together. The core question, which was to study miniaturization in the centipede genus. So based on this particular thing, whatever gymnastic that I did, also shed light on its integrative taxonomy and biogeography, as well as how old this lineage is, as well as what are the dynamics of body size and how did it change through evolutionary time. And this leads to a question, which is what are the drivers of patterns in body size evolution? And we can also ask if, as I mentioned earlier, does pedomorphosis as a phenomenon play a role in Asanada's miniaturization? We need to test that. And also, what are the signatures of reduced body size from a genomics point of view relative to what the other scolopendrid centipedes? And this is exactly are very tiny steps towards studying a lesser known taxa in a lesser known landscape. And this wouldn't have been possible without an enthusiastic team of researchers from our lab. And I would like to thank all of them. And I would like to thank Dr. Janna Vizoshi for providing me with the support and all the insightful information. I would also like to thank Evolution Meetings for providing me this opportunity to present my work in the Ernst Mayr Symposium. Thanks a lot.
Thank you. All right, and we have a minute or two for questions.
And if you could minimize your video
Meeting
panel to the
toolbar,
right, yeah, it'll make it easier if someone has a question about a specific figure.
but also feel free to use the chat tool as well. Okay, Maya has her hand raised. Jacob, can you unmute? Or I can. Oh,
Hi. Yeah,
Yeah.
Beanbar here. This is not a question, sorry. I just wanted reflecting what people were saying in the chat. If future presenters could minimize their meeting controls and pictures on the side, that would be wonderful. Sorry.
Okay, sorry for that.
Yeah, certainly not your fault. That was not an issue during our test run. So Zoom issues, we can all accommodate. We certainly just didn't want to interrupt you during your presentation. And so
We have
Um,
a question from Ulrich.
yeah, if it's a pretty quick one, and then we can get the next speaker prepared. So, Olivia, if you want to
Um,
get your slides here.
yeah, so my question would be maybe I didn't really maybe I missed a part or something. So does the miniaturized species only appear on islands, or do you also find it in other places?
No, so it's broadly present in peninsular India, but there are occurrences of these species of this genus being present in Africa as well as different island chains, as well as Papua New Guinea and Australia. So, but the only problem that is there is there's no evidence of these individuals being present. These are just pictures. These are just pictures uploaded by different people on iNaturalist or something like that.
Okay,
All right.
thank you very much.
Thank you so much. And with that, we have our next speaker, Olivia.
Sure, I'll go ahead and get started. Thank you
Mhm.
so much, Becky, and thank you all for being here today. My name is Alivia Nytko, and today I'm going to be talking about the evolutionary dynamics that have contributed to the existence of rarity through time. So, species exist in varying abundance, such that there are a few species that are often found in high abundance, but many more species that are found in lower abundance. And so, these species that are often found in high abundance are often considered to be common, while these species in lower abundances are often considered to be rare. However, this can vary across spatial scale. For example, a species can be widely abundant across the regional landscape, but be found relatively sparsely within a community. And vice versa, where a species can be widely abundant within a community, but be found relatively sparsely across the regional landscape. But rarity is more than just the abundance of species. There are also diverse dimensions of rarity that exist at both regional and local scales. For example, a species can be geographically rare at the regional and at the local scale. Here, the plus indicating rarity and the minus indicating commonness. And so at the regional scale, this might look like a species with a small range size, while at the local scale, this might look like a species that's found in low abundance. And so this is often what we think of when we think of rarity. However, species can also be functionally rare at both regional and at local scales. At the regional scale, this might look like a species that has a unique set of traits across a broader group of species, whereas at the local scale, this might look like a species that has a unique set of traits within a community. And finally, species can also be phylogenetically rare at the regional and local scale. At the regional scale, this might look like a species that's isolated across the broader tree of life, while at the local scale, this might look like a species that has high evolutionary distinctiveness within a community. And so throughout time, we've often used the abundance of species in order to categorize species as being either rare or common. And while we still use these abundance-based metrics today, more recent frameworks, such as the functional rarity framework and the EDGE protocol, have begun incorporating these diverse functional and phylogenetic dimensions of rarity. However, no system to date has yet incorporated all three geographic, functional, and phylogenetic dimensions of rarity at both regional and local scales. However, by considering all of these dimensions together, I've created a general model of discretized rarity, which includes 64 different ways a species can be rare, some of which are novel and some of which already exist. But importantly, because these are now all simplifications of the same general model, they can be compared directly in their ability to explain variation in biology. And therefore, the best definition of rarity becomes a statistical question rather than a matter of belief. For example, when we apply the general model of discretized rarity and its submodels to 1,011 British plant species, we find that the submodel that incorporates regional and local dimensions of geographic rarity, regional and local dimensions of functional rarity, and local dimensions of phylogenetic rarity performs the best in explaining 54% of the variation in distribution change and 45% of the variation in flowering phenology. And so not only did the application of this submodel show that rarity does indeed explain variation in biology, but also that dimensions of rarity interact, such that the singular effect of any one dimension depends on if other dimensions are also considered. And so we know that there are many ecological drivers of rarity. but we know much less about the evolutionary dynamics that have also contributed to the existence of rarity as a state or syndrome through time. However, there may be two interconnected processes contributing to its existence, these being diversification dynamics, specifically differential diversification, and evolutionary transitions or state changes through time. We already know that there's substantial evidence for heterogeneous diversification in angiosperms, and that much of this variation is associated with plant traits or syndromes of plant traits, such as floral symmetry and stamen number. We also know that there are certain traits, such as self-incompatibility, that persist via increased speciation, despite the short-term advantages of self-compatibility, which suggests that rarity may also be one such unfavorable character that persists via differential diversification. And if this is the case, we would expect to see that rare lineages have higher diversification rates than common lineages. However, it may be as we might expect that rare lineages have greater extinction relative to speciation. And instead, these rare lineages might persist as these ephemeral states that are driven by these repeated and rapid transitions from commonness to rarity. So using this new multidimensional rarity of species, we first asked what evolutionary processes have driven the existence of rarity through time, and how do these processes vary across the different rarity dimensions considered? Well, to do this, we first had to categorize the multidimensional rarity of 1,011 British plant species. To do this, I built an R package called GD Rarity, which implements the general model of discretized rarity. And so the way this R package works is it gathers range data, trait data, phylogenetic data, as well as user-specified abundance data. And it then uses these data in order to calculate the different dimensions of rarity, as well as determine which combination of dimensions best explains variation in a given biological process, in this case, it being distribution change and flowering phenology. And then once the optimal thresholds are determined, the top model is then used to categorize the species into their respective rarity types. So once the rarity of British species was defined, we then used univariate hidden state speciation and extinction models, or these HISSE models, to first see if the evolution of one single dimension of rarity was correlated with diversification processes, as well as to characterize the transitions between rarity and commonness. So this is just one example of a HISSE model in which it infers the speciation rate, the extinction rate, as well as the transition rates, once again, to and from rarity and commonness for any one single dimension of rarity. And from this, we were also able to calculate the net diversification rate, which is speciation minus extinction, as well as the turnover rate, which was speciation plus extinction. And so these values can be compared within rarity dimension in order to see how rare versus common lineages compare, as well as between rarity dimension in order to see how differently rare lineages compare. To also infer the same parameters across two dimensions of rarity simultaneously, we used bivariate multi-character hidden state speciation and extinction models, or more simply, these mu-HISSE models. And these work very similar to the HISSE models in the fact that they infer the speciation rate, the extinction rate, and the transition rates. However, now lineages can be common in two dimensions of rarity simultaneously, rare in two dimensions of rarity simultaneously, or intermediately rare, where they're rare in one dimension and common in another. And so similarly to these HISSE analyses, these diversification rates can be compared within models in order to understand how these intermediately rare lineages differ, as well as between models in order to understand how the different combination of rarity dimensions affects diversification. So is rarity better explained by these diversification dynamics or by evolutionary transitions? Well, the univariate rarity dimensions were correlated with diversification processes. However, rarity was actually associated with low or even negative net diversification. As we can see here on the y-axis, we have our different dimensions of rarity considered. Once again, the plus indicating rarity and the minus indicating commonness. And so for functional and phylogenetic dimensions of rarity, they exhibited negative net diversification, which basically means that extinction is outpacing speciation. And even for the rare lineages that did exhibit positive net diversification rates, they were always slower than the comparable common lineages. And so not only did rare lineages go extinct faster than they speciated, they also did so at much slower rates compared to common lineages. So there's not much support for existence or persistence by differential diversification for these univariate analyses. But what about those bivariate analyses or those mu-Hissy models? Well, given the univariate conclusions, we might expect that a lineage that is common in two dimensions of rarity simultaneously to have the highest diversification rate, while lineages that are rare in two dimensions of rarity to have the lowest diversification rate. Once again, though, that's not what we see. In fact, we see that rarity dimensions interact to affect diversification processes. Specifically, the combination of rarity and commonness in these intermediately rare lineages often resulted in either the highest diversification rate or the lowest diversification rates. So while it may be that certain combinations of rarity do persist via differential diversification, given the models tested here, rarity as a whole state or syndrome likely is not explained by diversification dynamics. But what about those evolutionary transitions? Well, transitions moving out of rarity were faster than transitions moving into rarity. However, transitions in either direction occurred relatively rapidly. And when we look at the average speciation rate, extinction rate, and transition rates in rare versus common lineages, the next most probable step for any rare lineage is to actually transition out of rarity, whereas the next most probable step for any common lineage is to speciate or to go extinct. And while rarity represented the most probable ancestral state for many of these different rarity dimensions, many of these same dimensions are also expected to occur in very low frequency at equilibrium. So while it's likely that the current frequency of multidimensional rarity today represent these non-equilibrium evolutionary dynamics, the persistence of rare lineages through time is better reflected given the rapid and repeated transitions from commonness rather than diversification dynamics. And so for a long time, we've defined rarity using abundance-based or geographic dimensions of rarity. However, there are diverse functional and phylogenetic dimensions that are not only important to defining what a rare species is, but they also interact to affect both ecological and evolutionary processes. And so for a long time, it's also been very difficult to directly compare these different multidimensional perspectives to rarity. However, we now can using the general model of discretized rarity, which finds the best definition of rarity based on its ability to explain a given biological process, once again, therefore making the best definition a matter of statistics rather than belief. And so rarity has often been conceptualized as a consequence of ecological processes and human disturbance. And while this is not untrue, there are also important evolutionary processes that have also contributed to its existence through time. So by using this new multidimensional approach to defining rarity, we show that while yes, rare species do go extinct, rare lineages are most often lost due to transitions to commonness rather than extinction. We also find that many of these different rarity dimensions are expected to occur in very low frequency at equilibrium, once again suggesting that the current prevalence of rarity today is likely a consequence of ecological factors and these non-equilibrium evolutionary dynamics. And so taken together, these results suggest that rarity has likely existed and will continue to exist as an ecologically important but ephemeral state that's largely driven by these repeated and rapid transitions from commonness to rarity rather than diversification processes. And with that, I'd like to thank all the many people, groups, and organizations that have helped with this project. I'd also like to thank SSB for hosting this Mayr Award Symposium. I've also included a link to the GD Rarity R package here, or a URL, in case you're interested in checking that out. And with that, thank you so much for listening, and I'd be more than happy to take any questions with the remaining time.
Awesome. Thank you. We do have time for a quick question if anybody has. Clayton, feel free. Jacob will unmute you. And then while we do that, Matthias, you would like to get your slide set up?
Hey, awesome, wonderful talk.
Thank you.
I was wondering, I know your research, a lot of this is kind of looking at evolutionary timeframes and lineages and stuff. One of the big benefits of the Rabinowitz framework is it gives us a framework for conservation right now. say with the Nature Serve and IUCN and things like that. I was wondering if your framework can be related to conservation today in that
Yes,
same way.
yes, for sure. Let me stop sharing real quick. Yes, totally. So the Rabinowitz has, you are totally correct. It has been very important for conservation. Um, the components of this framework, such as the functional and the phylogenetic aspects, there has also been a lot of work on why protecting and managing and conserving functional traits are so important, as well as conserving evolutionary history, which is often related to the functional uniqueness of species. So yes, it can be extended. And I write about that in the manuscript that's in review at American Naturalist right now. But that's great. And that's really the goal of this, is that it will be used in conservation as Rabinowitz is now. Thank you.
Awesome. Thank you, Olivia. All right. And we have now our last speaker of the session, one of two sessions for Mayr Award today. Right, Matheus, you're all up and ready. So with that, go ahead and take it away.
All right. Hope you guys are all seeing my slide here. Thank you, Becky. Thank you, everyone, for coming here. I'm really excited to be here in the symposium. Thanks for the folks inviting me over. And I think a lot of people will like this talk because we're going to discuss a lot of the topics that were talked about in other talks, like islands, non-equilibrium processes, and actually another important driver of biodiversity, if you ask me, which is extinction. So this is all related to my PhD work in the University of Michigan, supervised by Dan Roboski here. And I really want to emphasize the importance of extinction. If we were to graph biodiversity on Earth in this y-axis here, since the Phaerozoic, we would see a lot of ups and downs, moments in which biodiversity increased and decreased. And the big dips are these events called mass extinction events, moments that totally reshape the dominant organisms in the planet. Think, for instance, the extinction of the trilobites at the third mass extinction. But even though the fossil record tells us a lot about extinction, it's really hard to translate what we learned from the fossils into something that we can use or to understand the living biota, because the living biota represents just 1% of all species that have ever existed. And what we know about it, which is not much about extinction, is just that this biota is highly threatened. The IUCN counts more than 48,000 species threatened. That's 28% of the total. And this risk varies a lot between different groups of organisms. So I want to emphasize here some sort of gap between basically two types of extinction biology. One is centered on fossils. It usually has a global scope and is centered on extinction that happens at the million-year time scale. And the other, extinction biology, is more centered to population trends, usually with data collected at the local scale, and that reflects threats on the scale of 100 years or maybe a few decades. And what does lie in the between of all of this, right? The promise of studying extinction on some sort of mesoscale is that maybe it could be more generalizable, more meaningful to the living biota, but we still don't really know the scales in which this process would manifest. Would it be in the regional scale? In which phylogenetic scale would it be? So this is what my talk today is going to be about. I want to explore how extinction looks like at this intermediate scale, and in particular, how does it vary between geographic regions, between organismal groups, and between closely related species within these groups. And in order to do that, we need some sort of natural experiment of survivorship, something that reveals the potential to survive extinction that some species may have. And actually, we have this sort of condition here. This, what I'm going to show you in the next slide, is a GIF that shows the configuration of the planet since the last glacial maximum 25,000 years ago. The colors that are important to us, they show temperature. What I want you guys to focus are the black lines that show the coastlines of every land mass through time. And in particular, I want you to pay attention to the Sahul supercontinent. This land mass that cover what we call today Papua New Guinea, Australia, and Tasmania here. So as the glaciers melted and the sea levels rose, all these coastlines retreated, fragmenting this landscape and stranding organisms in these islands. And actually, Ernst Mayr himself noted, perhaps during his fieldwork in Papua New Guinea or perhaps later in his life, that once these islands became isolated from the continent, they would gradually lose species as the local populations inside the island would become smaller due to less resources, and the rescue effects that would keep these subpopulations would decline. So here, for example, this is a map of some of the land bridges in Papua New Guinea from Mayr and Diamond's book on birds of the northern Melanesia. So to model this, I basically assumed that the local extirpation rate of any given species within an area would be a function of the island area. And in particular, I modeled this as an exponential distribution that is assumed to affect all species, but the shape of this curve, how strong is the relationship, is species specific. In particular, we are talking about a parameter that would tell the curvature of this effect between area and extirpation rates. You can think of this as a surrogate for the minimum viable area of a species, because in islands smaller than this area, this local extirpation rate should be very, very fast. And how fast are we talking about? Well, this is the second parameter here, which is basically some sort of extinction delta, something that is capturing the maximum speed into which extirpation can happen in any given island. These are the two traits that we are comparing today: the speed, the temporal component of persistence, and space, how sensitive to space change that species is. So now let's come back to Australia here, and in particular to Tasmania. If you look at Google Earth in Tasmania right now, you'll see a lot of islands with all different sizes that I colored here with these different colors here, colder colors meaning bigger islands. But this actually, this archipelago is associated with a rich history in which all these land masses were previously connected to each other or to different subsets of these land masses, and their areas also vary through time. We can be a little sure about this now because now we have good data on the coastline composition at any given point, like at intervals of 500 years since the last glacial. And if we add some species data into this, as presences and absences in which islands, I can use algorithms from comparative method to triangulate the patterns that I'm interested about. So for instance, this would be the data set associated with a single species, in this case, the eastern quoll. And from that, but we didn't work with just one species, right? We compiled actually a lot of species occurrences from this large database that the government of Australia keeps. It's called the Atlas of Living Australia, and it compiles historical field collections and also community science data. It's all created by I further cleaned this data by removing typos, errors, invasive species, any flying or organism that was capable of oceanic dispersal. I did this for amphibians, for squamate reptiles, and for mammals. And I was very happy that I could include organisms for at least these 8 ecoregions in Australia here that I represented with these polygons. And they capture a lot of different environmental gradients, not only like different faunas and biotas in general, but also some abiotic changes too. So a quick recap, we're interested into studying the shape of the distribution here. We can capture that with 2 parameters, and we did this for all these species in my final data set here. 72 species of amphibians, 352 species of squamate reptiles, and 46 species of mammals. This is my final data set after I removed all the species that were actually best fit by some sort of equilibrium model of metapopulation or island biogeography dynamics, and the species that also fit a new model in which the past history of fragmentation had nothing to do with the current distribution of species in islands. But coming back to the ones that we care about, the ones that fit our natural experiments, we can use a linear mixing model to separate the effects related to ecological context or ecoregion and as a random intercept, and we can extract the species traits as a fixed effect in this model here. Finally, I also downloaded the data on body mass, mostly mass in grams, from this database from Moore et al., and I used PGLS to compare these variables here. So now a quick description of the results here. Both components, the space and time components of persistence, vary by at least 3 orders of magnitudes among different species in Australia here, as you can see from these scatter plots. But this is the raw estimates, right? We have to extract the geographic signature of this, and I did that with the linear mixing model. So in this next slide here, what I'm going to show is just one of the components, how sensitive to area change every species is. And I'll show this as a polygon. And the warmer colors will mean that in that region, that organism is more threatened, or the persistence there is more sensitive to area change. And the colder colors will be the opposite. Those will be the areas that somehow promote persistence, and the animals require less area to persist long-term in these land bridge islands here. So when we plot the amphibian data set, we don't see a strong geographic trend, but we do see one exception in the northern savanna region. I'm still trying to understand if these islands have a specific feature that promotes persistence for this, or if this is some sort of like outlier that doesn't have a very specific explanation. Now, the mammal parents are a little easier to explain. Basically, all the savanna ecoregions are associated with less persistence for mammals. The exception should be the Pulberia shrublands in the West Australia here, but that community, at least in my data set, is highly enriched in herbivore and small omnivore mammals. So maybe this geographical effect is actually the effect of the pool of the species that is being perceived as the regional effect. That would explain this outlier here, but the rest, like the savanna pattern, is a little strong, and this emerges from the mixed model, right? We're not doing this explicitly. Another organism with a latitudinal trend are the squamid reptiles. They also have a latitudinal trend, but it looks like reptiles need more areas to persist in the colder, more south regions of Australia, while they mean less area in the savanna associated ecoregions over there. Now I want to quickly dip into the trait analysis here. We are comparing body mass and the two components of persistence here. This is the sample size of every analysis. And I'll just like drop the results pretty quickly in your screen here. So basically, what the PGLSs show is that large mammals and large amphibians both get extirpated faster in islands. But this is not mediated by a sensibility to space. It's just like an overall rate that is faster. The large squamids, on the other hand, persist better, not only because their maximum extirpation rate is lower, but also because they are less sensitive to area changes. So coming back to the questions line here, how does extinction look like this mesoscale, right? Well, I think the geographic and the biological variation in persistence are kind of like mixed together. They're intertwined. Like, even though we see no obvious trend for amphibians, we can clearly see some latitudinal trends in persistence in both mammals and squamid reptiles. And at shallower phylogenetic scales, this is mediated by body size. And of course, this raises questions about other traits that could also be mediating persistence. And we can think on a long list of traits like trophic level, habitat requirements, life history, parthenogenesis, viviparity for squamids, and many others. So it's very exciting to be able to think that maybe in the close future, we will be able to explore this hypothesis here. And speaking of future, I actually want to quickly discuss here some future improvements all this approach can have. Of course, we have to complete the trait data set. I had to drop half of my amphibian species from the body size analysis. But so we want to complete that. We also want to account for imperfect detection. I know this very well. This is a big component of my other paper on persistence that we published last year in Ecology Letters. We need to be sure that these absences are actually not failure to detect a species, especially in tiny islands. But I also want to quickly discuss here some fundamental limits of the approach that I'm trying here. We cannot study the species that only occur in the mainland. We don't know if they were in the islands before. We cannot right now tease apart any sampling that is specific to a region to the effect of the region itself. This could explain some outliers maybe, but now we cannot really be sure. I need to explore other ways to maybe tease apart these two things. But another big fundamental limit is still time scale, right? Land bridge islands allow us to see how extinction look like in the 1,000-year time scale for the first time, but there's still a major gap in the 100,000 years time scale in which we still don't know how extinction looks like. And yet, I think we have to try to push a research program on extinction biology, because right now we are changing our plans and we need to know. For instance, this is the map of fragmentation in the Amazon here showing red, and we need to know the consequences to biota of this large-scale environmental change here. And of course, we need to know how persistence look like in other organismal groups too. That's why I think it's exciting to at least think that now we can have another tool to look at extinction here. And with that, I am finishing my talk here. I want to thank everyone that mentored me, all the institutions that funded me, my 2 labs, everyone that contributed to these large databases that I used. And now I can take any questions you may have. This is how you can find me, my email. And thank you very much for your attention. Thank you.
Yes, for a wonderful talk. Um, There's only a minute left, so if someone has one short question, you are welcome to ask or type it in quick. But this is also the last talk of the first session of the Mayr Symposium. There'll be a quick 30-minute break, and then we will pick up again for session 2, where we'll have another 6 great talks. So with that, I'd just like to first thank all of the speakers for sharing your incredible science. It's amazing what you guys have been able to accomplish during your dissertations. And thank you everyone here for attending and supporting our early career scientists. So have a good afternoon, evening, early morning, whatever time zone you are in. Really good job, speakers.